So, when we started the discussion, I have showed you the subtopics in anatomy. So, the subtopics in anatomy were upper limb, lower limb, thorax, abdomen and pelvis, head and neck, and neuroanatomy. And I have told you that these all together makes the gross anatomy. And what is meant by gross anatomy, I have told you that gross anatomy is the study which you are going to do in the dissection hall. That means gross anatomy simply means the study of anatomy which does not require a magnifying lens or which does not require a microscope. Those anatomy which you can see by your naked eyes. Okay, so when you dissect, what you are going to see is through your naked eyes. You doesn't need a microscope there. So the anatomy of upper limb, lower limb, thorax, abdomen, pelvis, head and neck, and neuroanatomy, all this is comes under gross anatomy. So these are the things you are going to do here in your dissection hall. Or this is the anatomy where you does not need a microscope. You can see it with your naked eyes. So we have discussed. So whether you are dissecting upper limb or you are dissecting lower limb or you are going to dissect the brain or you are going to dissect the abdomen, the things you are going to come across is one is blood vessels, one is muscles, one is nerves, one is the lymphatic supply, one is the bones. So we have discussed in detail about each and every of these. Okay, so that's why I have discussed about blood vessels, nerves, lymph supply, muscles and bones, because even regardless of which part of body you are going to dissect, these are the things you are going to come across. So we need to know the basics. So now we know the basics of gross anatomy. So we have completed the basics of gross anatomy. Okay. And also we have completed the basics of osteology, right? We have completed the basics of osteology. We have discussed in short about the different bones in human body. So we have also completed the basics of osteology. Now, what is remaining is general anatomy, histology, embryology, and radioanatomy. So these are the remaining topics. So the remaining topics is one is general anatomy, one is histology, one is embryology, and one is radioanatomy. So these are the remaining subtopics in anatomy. So now just let's know the basics of this also. Now let's know the basics of this also. So now let's come on to, so this all we have discussed. All of this we have discussed. Now let's come on to the remaining topics. One is histology. So histology is basically, I have told you that histology is the study of tissues. Histology is basically study of tissues. So I have told you that in your first year MBBS, how your normal day will be. You will be having your classes start. What's followed in Calicut Medical College, and I think this is the same pattern followed all across the world. So your classes will be starting at 8 o'clock, and from 8 o'clock to 10 o'clock you will be having a theory session of 2 subjects or 2 sessions of 1 hour each. Okay, after 10 o'clock, from 10 to 1 you will be having dissection. Okay, so that's the 3 hours spent for dissection, and after that you will be having break for 1 hour. So 1 to 2 will be break, and after that you will be having lab. Okay, from 2 to 4 you will be having lab. So your curriculum is between 8 and 4 usually. So this is how it's followed. So in the lab, you have anatomy lab, physiology lab, and biochemistry lab. So for anatomy, gross anatomy, we have already done in the dissection part. So 10 to 1 is where you learn the gross anatomy. So what we learn in anatomy is actually histology. So anatomy lab, what you're going to learn is actually histology. So in histology, you will be having different types of tissues. You will be given different slides with different tissues. And we are going to observe it under the microscope. And we are going to observe it under the microscope and identify which type of tissue it is. Okay, so here we cannot do it with our naked eye. We need a microscope here. So that's why study of tissue. The other study that we do in dissection, or that was called gross anatomy. We have discussed about it. Now what we do in the anatomy practical lab. What we do in anatomy practical lab is actually histology. We are learning about tissues by looking it under the microscope. We will be given different slides. We will observe it under the microscope and we will identify which type of tissue is this. Okay, so here I have shown you certain tissues here. So this is the tissue of bronchus, this is of esophagus, this is stomach, duodenum, colon and all. So you will learn the difference between each and every one of these. Okay, you will learn the difference between each and every one of these. Now at this point, each of this image may be looking similar for you. At this point, you may be, for example, you might not appreciate the difference between stomach and duodenum, or you might not be able to appreciate the difference between liver and pancreas. But believe me, when you enter the medical school, you will be taught how to differentiate between these tissues. Okay, so you will be taught how to differentiate between these tissues. So that's basically what we do in the histology lab, or that is the histology part of anatomy, studying the microscopic structure. Okay, now I'm just showing you this is the histology structure. Or if you take a section of your testis, okay, if you take a section of your testis, this is how it looks like. This is how it looks like. Okay, so this is actually the histology or the microscopic appearance of testis. So you can see that here we have got Leydig cells, we have got Sertoli cells, we have got spermatogonia. Okay, you know all of these terms. Okay, so we have Leydig cells, we have Sertoli cells, we have spermatogonia. So in any histology tissue or in any histology section, what happens is that there are usually predominantly two colors. Okay, there are predominantly two colors. One will be a pink color, one will be a pink color, and second one will be a violet color. And second one will be a violet color. Now remember that this pink color is usually the color of cytoplasm. So cytoplasm usually takes pink color, and nucleus usually takes violet color. So in this figure you can see that this part and all, these all are pink. So these all are the cytoplasm, but some part it is violet color. So these all are the nucleus. So these all are the nucleus, right? So usually in any tissue, this is the basics of histology. This is the basic of histology. Usually in any tissue, cytoplasm is stained pink in color, and the nucleus is stained violet in color. So I hope it's clear to everyone. So in any histological tissue, we have two colors. Every tissue takes two colors. The cytoplasm usually takes the pink color, and the nucleus usually takes the violet color. And therefore, you have to draw these figures, right? So you have to draw these figures. So whatever you see under the microscope, you have to draw it in your record. Okay, whatever you see under the microscope. So if you're seeing this section of a testis, so if you're seeing the microscopic section of a testis, you have to draw the microscopic section of testis inside your laboratory, inside your practical record. Okay, you have to draw it in your record. So for drawing that, we'll be needing two types of color. Which all are they? One will be pink and second one will violet. One will be pink and second one will be the violet. So this pink color pencil is called eosin. This pink color pencil is called eosin. So in your first day itself, you will be buying these two pencils. These two pencils are essential. Okay, you will be buying these two pencils. This pink color is called eosin, and this blue color or violet color is called hematoxylin. Violet color is called hematoxylin, and blue color is called, or violet color is called hematoxylin, and pink color is called eosin. Okay, so hematoxylin is violet color, eosin is pink color, and we also know that violet color is usually taken up by the nucleus. So nucleus stains violet in color. So with hematoxylin, you are going to draw the nucleus, and you know that the cytoplasm stains pink in color. So I use the eosin pencil to draw cytoplasm, and I'll use the hematoxylin pencil to draw the nucleus. So if I'm asked to draw the section of testis, I'll take my pink color and I'll take my blue color. Sorry, I'll take my violet color. So I'll just show you if I have pink here. So this is how I will draw. Okay, I can't actually draw it here. I actually need a pencil. So this will be how it looking. My cytoplasm will be looking like this. Inside my cytoplasm, I will be having. So this is my cytoplasm. I have used my eosin pencil to draw this cytoplasm. Now I am using my hematoxylin pencil to show my nucleus. So this is how it will be looking. You cannot see the proper demarcation at all when you see the actual tissue. So this will be how looking. It will be a pinkish background. It will be a pinkish background because cytoplasm is larger area. So it will be looking like this. So there will be a pinkish background. So I'll be drawing the pink color with my eosin, which stains pink in color, which stains the cytoplasm. And I'll draw my nucleus with hematoxylin pencil. I'll draw my nucleus with hematoxylin pencil, which is violet in color. I hope it's clear. So this is what we learn in histology lab. So these are the two pencils you have to buy before your histology lab. Okay. So that's about histology, and that's how we use the hematoxylin and eosin pencil to draw our records. Okay. Now I'll just show you the dissection kit. So along with hematoxylin and eosin, you are also going to buy a dissection kit in your first day itself. because you need dissection kit to dissect your cadaver. Okay. So this is actually an elaborated dissection kit. Okay, this is an extensive dissection kit. Will not be having all of these. Actually, what is essential in a dissection kit is three things. One is this. One is this. This is called a scalpel. This is called a scalpel. So you can see that I have got a handle for a scalpel, and at the end of the scalpel I have got a blade. So for piercing or to make incisions, I need scalpel. So this is one essential thing in your dissection kit. Scalpel. And other than that, I have two types of forceps in dissection kit. I have two types of forceps. So this is called a non-toothed forceps. This is called a non-toothed forceps, because at the end I can see that there is no tooth. At the end there is no tooth. Okay, I'll magnify it further. You can see that there is no tooth here. Okay, there is no tooth here. Okay, there is no tooth here. So this is my second type of forceps, and this is called my second type of forceps, and this is called a toothed forceps. So at the end of a toothed forceps, it's actually not visible in this figure. I'll show you. I'll just draw it for you. So if it's a non-toothed forceps, it will be like this. If it's a non-toothed forceps, it will be like this. Okay, so this is a non-toothed forceps. And if it's a toothed forceps, at the end I will be having certain tooths like this. At the end I will be having certain tooths like this. So to hold thick structures, so if you have to hold skin, basically you know that we use forceps to hold certain structures. So to hold tough structures like skin, you cannot hold it with non-toothed forceps. Okay, you cannot hold it with non-toothed forceps. It gets slipped away. Okay, so if you have a soft end at the end of your forceps, you cannot hold tough structures like skin. It gets slipped away. So for that purpose, we need toothed forceps. So for that purpose, we need toothed forceps. We cannot use non-toothed forceps there. Now, what's the purpose of non-toothed forceps? For example, you are going to hold a blood vessel. If you are going to hold a vein, okay, if you are going to hold a vein, you cannot use a toothed forceps there because it's sharp structure. Okay, it's sharp, and the vein is a soft, delicate structure, and those delicate structure gets pierced or it gets collapsed if you use the toothed forceps there. So we have a non-toothed forceps and we have a toothed forceps. So toothed forceps, this is a toothed forceps. So for holding tough structures or thick structures, I will use a toothed forceps, and for holding delicate structures like blood vessels. So example for a tough structure is skin, and example for a delicate structure is a blood vessel. So for holding a delicate structure, I will use a non-toothed forceps because the tooths are sharp. So if the sharp I'm using the sharp toothed forceps, it will pierce the blood vessel or it will pierce that delicate structure. On the other hand, I cannot hold a tough structure with a soft forceps. Okay, for example, a tough structure like skin or a tough structure like muscle, I cannot hold it with a soft forceps. For that, I need tooths. Okay, so for that I need tooths. So I'll be using toothed forceps to hold tough structures. So these are the three important. So you have many scissors, you have forceps, you have different types of forceps, you have scissors and all. But usually what we have in dissection kit, what are essential in dissection kit? One is scalpel to make incisions. The second one is a toothed forceps. Toothed forceps is for holding tough structures. And the third one is a non-toothed forceps. Non-toothed forceps is for holding a delicate structure. So these are the three essential things in dissection kit. Okay, other than that, you can have scissors, you can have gloves, you can have certain different types of tissue holding forceps and all. But these are the essential three things in a dissection kit, which you should definitely have before starting dissection. Okay, now that's a famous comedy which passes on in the successive generations in our medical college, that when we enter in our first year MBBS, our seniors will come to us and they'll ask that whether you have bought your dissection kit. So yeah, we'll say, yeah, I have got my dissection kit, I have got my scalpel, I have got my toothed forceps, I have got my non-toothed forceps and all. I'll say like that. Then they'll ask, have you bought anatomical snuff box? They'll ask you, have you bought anatomical snuff box? So we'll be just wondered, what is anatomical snuff box? So we'll think that we have forgot to buy anatomical snuff box and we'll again go to the store and ask for anatomical snuff box. So actually anatomical snuff box is not a tool used for dissection. Anatomical snuff box is a anatomical area or anatomical triangle in the part of your hand, in the hand. So this is my anatomical snuff box. Okay, this is called anatomical snuff box. So in this figure I can show you. You can see that in this figure these are certain tendons of different muscles. Actually, those are the tendons of abductor pollicis longus, extensor pollicis brevis, and extensor pollicis longus. Don't be, don't get shocked with those terms. You are also going to say it like this. Okay, after just 6 months, you will be also saying it so fluently. Okay, so I have two tendons here. So these tendons makes a box here, or it makes a triangle here. This triangle is actually called anatomical snuff box. This triangle is called anatomical. This triangle is called anatomical snuff box. Okay, so our usual seniors just want to have fun, so that's why they just want to irritate us, so that's why they'll send us back to the store to buy anatomical snuff box, and we don't know anything. We'll go and ask Indian store to give us anatomical snuff box. Only then we'll come to know what actually is an anatomical snuff box is. So anatomical snuff box is not a thing, it's not a box, it's actually a part of your body. In your hand you have a triangular area formed by different tendons, formed by 3 tendons mainly, and that is called anatomical snuff box. So I just wanted to mention it here. So in the dissection kit, you are going to have, essentially you are going to have 3 things. One is a scalpel to make incision, second one is a toothed forceps to hold tough structures, and the third one is a non-toothed forceps to hold delicate structures. I hope it's clear. Okay, now moving on. So we have spoken about histology. Now the next remaining thing was embryology. So embryology means it basically the word itself contains the meaning. The word itself contains the meaning. Embryo. Embryo means, we know what an embryo is. Embryo is the baby before delivery. Okay, in simple words, embryo is the baby before delivery. That means after fertilization, before delivery, we call the baby as, or we call it as the embryo. Okay, so this embryo, the study of embryo, okay, study of this embryo. So in the initial stages we call it as embryo, not till the delivery. In the initial stages we call it as embryo. Okay, so the study of embryo is called embryology. So in this figure you can see this is just a pictorial representation of what we are going to learn in embryology. This is just a pictorial representation of what we are going to learn in embryology. Okay, so here the fertilization is occurring. You can see that a sperm, this is a sperm, right? This is sperm. This is sperm. This sperm is going to pierce this ovum. So this is a sperm. This is a ovum. And sperm and ovum has fused to form the embryo. Right? Sperm and ovum has fused to form the embryo. Now embryo has got different cell stages. It has got morula. It has got blastula. Okay, there are different cell stages. You can see that it is like this. So embryo has formed. Now embryo will be having different cell stages. Okay. Now after that, you can see that this embryo is now gaining, more or less it's now looking like a proper baby. Okay, so it started to develop head. It started, so you can see that this embryo has started to develop head. It has started to develop head. It has started to develop limbs. Okay, it has started to develop lower limbs also. Okay, so that's now the embryo has developed further. Okay, you can see that the limbs are very well developed here. Okay, not completely, but it is more developed. Head is more developed. You can see the eyes have come. Okay, now this is a completely developed baby just before delivery. So that's basically what we learn in embryology. So from the point of fertilization of a sperm to ovum, to an embryo, to different cell stages of an embryo, to a more or less similarly looking like to an human baby, to a proper baby. So this is what we learn in embryology. So for different structures, this is just the pictorial representation, or this is just the most simple pictorial representation of embryology. In embryology proper, you will be learning the development of each and every structure. You will be learning the development of heart, you will be learning the development of brain, you will be learning development of kidney, you will be learning development of placenta. Okay, you will be learning development of each and every structure. So in embryology, basically this is the thing that you have to learn. This is the thing you have to learn. You have to learn the study of embryology, simply the study of embryos. Okay, now this I just want to show you a figure. This is actually development of heart, guys. This is actually development of heart. So this is just an example. So what is embryology? I have shown you in this figure. Okay, the sperm and ovum fertilizes to form a proper baby. That is embryology. Now for each structure there is embryology. So for the heart, this is the development of heart. So you can see that there are actually 2 heart tubes here, or we call it as the endocardial tubes. Okay, we call it the endocardial tubes or heart tubes. So these heart tubes fuse together. Okay, these heart tubes fuse together, and you can see that this is I'm not explaining these stages because at this point you can't understand it. Before learning it you have to know certain terms, but just see that this is the way you study in embryology. So I have got two heart tubes. These heart tubes now got fused. Now this heart tube is differentiating into different parts called truncus arteriosus, bulbus cordis and all. Now you can see at the end it forms the proper structure of a heart. Okay, so that's how embryology works. So from the beginning, so at the 20 days of fetal life, or at least 20 days of life of the embryo, there will be two separate heart tubes. At 21 days it starts to fuse. Okay, at 22 days it's less differentiated. At 23 days it becomes more and more differentiated. By 35 days we can see that it is more or less likely to be heart. Okay, it is more or less similar to a adult heart. Okay, so this is just the development of heart. So similar to this, you have to learn the development of kidneys, development of lungs, development of diaphragm, development of placenta and all. So I just wanted to show how you are going to learn in embryology. Okay, we are not going into detail, but you just wanted to know when you get the book of embryology, you should have an idea what you are going to find inside that book. Okay, so that's about embryology. So we have discussed about embryology, what we are going to learn in embryology with an example. Okay.